Welcome and good day. Today is the day for the pitch. My name is Anthony Viscogliosi, and I will be your moderator for tonight's program, a very interesting program we have for you. As a reminder, this is a monthly program, and it's scheduled for broadcast on the second Thursday of every month at 7 p.m. Eastern Time. We hope to see you at each and every broadcast. Each program will present two promising companies. Those companies will communicate their technology, their business plan, and then our panel will interrogate the companies to understand their technology, their plan, and what opportunities lie ahead for them. To begin, it's my great pleasure to introduce to you our panel. And let me first introduce to you Audrey Beckham. She is an independent business consultant today with a substantial history in the medical device industry as a leader. In her executive roles in the industry, she has been in product development, professional education, and quality remediation, leading all of these areas. Next, it's my great delight to introduce to you Michael Sherman. Mike brings more than 30 years of experience in the medical device industry, specifically in the orthopedic technology field. At one point, he was the largest patent holder at Sophomore Danek. He has 20 years in the industry and more than 9 years in venture capital. Welcome, Mike. And thirdly, we have Dr. Scott Bruder, an MD and a PhD. Scott brings more than 30 years of experience as well. Scott's experience is first as a clinician, then as a scientist, then as an operator, as a chief technology officer, in fact. Recently, Scott has focused in being a leader in product innovation and product development through his consulting franchise in the areas particularly of biotechnology, cell therapy, and diagnostics. As for me, I've spent my career in healthcare and finance. As an entrepreneur, as a founder, as an executive, as a financier. Along with my two brothers, Mark and John, we operate the single family office of Viscogliosi Brothers. What other name could it be? In that franchise, we have founded, invested in, and built more than 35 neuromusculoskeletal businesses. And we've created 15 private capital funds to invest in those businesses. Well, I've had the delight to serve as an innovator, as a chairman, an executive chairman, a CEO, president, managing director, and chief cook and bottle washer. It takes both hands to make anything happen in this field, but it takes a great mind, great talent, and this is what we look for in the opportunities that we have today. Great innovation, great minds, great talent, and great dedication. And now, it's my pleasure to describe to you and introduce to you our next company for the pitch. Now, this company deals with a very serious and costly healthcare problem. Not only a healthcare problem in orthopedics, but a healthcare problem in nearly all fields globally. Infection control. What if there was a way to make every single implant not have any microbial infection associated with its implantation and its permanence in the patient? Eliminating this risk is one of the holy grails in healthcare. Our next company is OrthoBond, and their goal is to do just what I've described. Let's run the video.
If you look at innovation throughout history, some of the things we think are so commonplace took time to adapt and incorporate. Think of something simple as washing your hands before surgery. You don't think about eating without washing your hands, much less doing surgery. But it took a decade or more for the data to build and for people to be convinced. So you think about that, it's a tremendous impact. That's what makes this fun for OrthoBond. This is one of the game changers.
The problem of microbial contamination is the broadest and largest problem really in all of healthcare. Each year there's millions of healthcare-associated infections occurring on implantable devices.
There's just a high percentage of implants that are contaminated. What OrthoBond has come up with is a permanent nanosurface that uses no drugs, no heavy metals, and actually kills bacteria. And that's the difference. That's our proprietary technology.
OrthoBond has a world-class team of scientists and engineers, and we came together a decade ago. What we realized was that to make a surface to kill microbes, you need a molecule which can attack the bacteria from the outside in. We tried a class of molecules called quats, found in nature, that carry a very strong, permanent positive charge. We've even tested it against mold and viruses, and we found that all of these different types of organisms have a net negative charge, opposites attract, and then once there, we do multiple things to attack bacteria, but it's perfectly safe to the patient.
Currently, we have nothing like it available to us in medicine. This is not a coating, this is a treatment to the surface of an implant so that no bacteria can attach to it.
Having such a unique treatment means we've tested our technology rigorously for both safety and effectiveness. The potential impact is huge. Doctors can go into surgeries without having to worry about patients getting infections.
What's exciting about this is going to be a platform technology. We don't desire to be a medical device company. There's plenty of companies out there. What we want to do is make all their implants better, cleaner and safer.
The technology is paradigm shifting. That is why I'm here at OrthoBond.
It's been a lot of hard work. This team has solved problems that no one else in the world could solve.
There's no other company in the world that does what we do.
The most exciting thing about the technology is that we can really apply it to virtually any surface.
If we can be moving beyond just medical devices to high-touch surfaces all over the world, we could potentially help prevent the transmission of disease.
Antimicrobial services can save lives.
I feel great pride because I know it's just gonna bring a lot of help for people who suffer through pain because of an infection.
We can make the world a safer place.
For me, it's intensely personal to think that something that I worked on is going to impact patients' lives, and this is just the beginning.
I see this as the next wave of new technology.
I think this is a game changer. We can give to patients throughout the world their best chance of healing.
Welcome back, panelists. I have one word to say: wow. Wow to what the potential is that OrthoBond is looking at and examining for great success. Presenting on behalf of OrthoBond is David Nichols. I'd like to sing his praises just for a moment if I can. I've had the honor to know David for more than 25 years. I've seen him grow up in the industry to become the leader that he is. When you mix great technology with great people, you have a real potential for great outcome. And so with that, David, welcome to the pitch. We're delighted to have you here. Let's turn this over to our panel for some questions. So Audrey, let's turn it over to you to begin.
Thanks, Tony. Happy to have the first shot here. Not in a bad way, a good way. So David, I'm really interested. I'm a mechanical engineer, so I just have to ask, how does this coating or this surface treatment get applied? What effect does it have on other surface treatments or coatings? The bone ingrowth capability, the bone cement adherence, potentially. So I understand it's a covalent bond, but can you tell us a little bit more about it?
Yeah, good question. And I'm a mechanical engineer as well, so I'm learning a lot of chemistry and microbiology through this project. But it's actually uh, really interesting. The base technology came out of Princeton, and it was for another application, as most things are. But they found a way to bond to the oxide layer of materials, so chromium oxide to titanium oxide. And how it's done is there's a monolayer of phosphonates that's put down. It's about 4 nanometers, and then that's the first step of the treatment process. And then on top of that, it is functionalized so that we can to create a handle that we can attach a molecule to it. So and that molecule that we attach is a quaternary ammonium. And so we bond that to it. And once we bond that to it, it's 40 nanometers is the thickness of it. So, a billionth of an inch. So it's almost imperceptible to it. And the logo that you saw in our video is what it looks like. It looks like a tree. So you're bonding to the oxide layer, and it sprouts up into this quat. And quats are very safe. Quats are in mouthwash, they're in shampoo, and this quat is safe to the human body, but it's not safe for bacteria, mold, virus. Those have a negative charge. We've got a positive charge, and so that's basically the interaction that this of how this works is actually killing, in this case, killing the bacteria. So it works very well with the metals that we use in orthopedics, which is where we started this technology. So it's actually, for a mechanical engineer, I understand it because it's a rupture. It's a mechanical disruption of the bacteria.
Great, thank you. And what kind of studies have you done to show the safety and effectiveness of the treatment?
We've been doing an extensive amount over the past few years. We've been working closely with the FDA to line out what safety and efficacy we need. And before I started there, that was the first test I did was, do we have a regulatory pathway? We've all seen companies with great ideas that don't make it through the regulatory pathway. And so we've been working so I checked this out extensively. And then we've been working with the FDA to line out what do they need to show that. So on the safety part, the biggest things, we had 2 large animal models, which Celeste could talk you through how we did those, but 2 large animal models to show that whether it was treated or not treated, there was no effect on the local tissue, the organs, the effect of the device, because the FDA's concern is the device going to change its function because you put this on there. And we did large animal models. We did the normal ISO testing, toxicity testing, ISO biocompatibility test for safety to prove that doesn't hurt anything. And then on the efficacy, there's a lot of normal assays that we can do to show that it kills bacteria, which is what we're using in this case. Last year during COVID, we applied it to some stainless steel surfaces and showed that it could rupture the common strains of COVID. So it can be applied to almost any material, polymer or metals.
Great, thank you.
Audrey, thank you for your questions. David, thank you for your answers. Because we're in the science part of the discussion, I'm going to go to our scientist, Dr. Scott Bruder, to invite you to go deeper in which areas you think best to cover to understand this technology and then market opportunity.
Thank you, Tony. And I think we all have had a sufficient level of scientific rigor, and so it's fascinating to hear the different questions that my panelists and you both ask. But David, is really very interesting. I have a couple of really short questions with hopefully short answers. So are you assuring that this is bactericidal and not bacteriostatic?
I'm the mechanical engineer, so explain that a little deeper.
Okay, so bactericidal means you absolutely kill the bacteria. So it sounds like that if you lyse them. Bacteriostatic means you just stop them from growing. further.
Got it. Yeah, so there's three mechanisms of action. Because of the charge, it actually attracts it. It's not like a hydrophobic surface that's going to push it away. So it actually attracts it. It actually pokes holes in it, so they actually leak out, so to speak, and then it ruptures the cell. So we've got great SEM images of just destroying the cells once they land on the surface.
And so do you have some commentary about the formation of biofilm on the surface? Because did I take your question, Mike? That's see, you got a couple of scientists here. That's really the problem, is biofilm. And because then that harbors, you've got a 4 nanometer layer there of your additive. But once you get, another 2 nanometers of biofilm, what's underneath it doesn't really matter. So that's a potential problem. If you've solved it, that's fantastic.
Well, I'll talk to this, and I'll let Celeste talk to this. What the data that we've shown the FDA, because the FDA wanted us to convince them about the bacteria that comes in contact with the implant during the operating room. And so we want to make sure that surface is clean to help prevent the formation of the biofilm. So the amount of bacteria that lands on an implant, especially the non-sterile ones like spinal implants, but even sterile packaged implants, sutures and anchors and joints, they get bacteria on them, which we believe becomes that start that create that film on that implant as it goes in. And the data that the presence of an implant in bacteria, it takes 1,000 times less bacteria to cause the infection because of the presence of the implant. So our goal is to keep it clean from the time it opens the package, stays on the Mayo, gets put on the instrument, and gets put in the body.
Okay, so let me turn to one more point then, because you talked about your extensive interaction with the agency. Do you think that you're going to be able to make a claim beyond reduction of bacterial colonization? Are you going to be able to make a claim of reduction of infection? Because I've been involved in some of that work, and they are 10,000 agencies looking for 10,000 patients in studies like that.
Yeah, it's a good question. We are wanting to ensure that the implant goes in clean. So we are ensuring that it's we can't say bacteria free, but it's 99.99% from the time it opens the package and goes in the patient. We're not going to be making any claims about reducing infection. We're going to be making claims about reducing the microbial contamination on the implant.
So it's listen, so I think it's a fantastic approach. My angst for you is that if you're not able to make clinical claims, that could limit your uptake, because there are some other considerations that companies have and physicians and payers have about saying, well, you're from the opening of the peeling package to getting into the patient, you're reducing infection. Yeah, that for sure, that's helpful, but ultimately it's about clinical outcomes. So I wish you a lot of luck. I mean, it's a fantastic approach.
Yeah, I think that's a good point. But if you go in spine cases today, see what doctors are doing to protect their patients by pouring vancomycin in the wound without any data. We believe this from talking to clinicians that are cleaning their implants before they put them in, that are washing the wound and flushing. They're very excited to have something I can tell them that I know when this goes in your body that it's going to be clean.
Congratulations, because I think it's got applications well beyond orthopedic implants.
Scott, thank you very much, and thanks for the depth of the questioning. David, you had suggested that maybe Celeste might be able to add a little bit of better understanding. Are there any comments that you'd like Celeste to make before we go on to Mike?
Sure. Celeste has done a lot of this research with Dr. Frank Camise at HSS. So you might want to comment on the work you've done.
I think Scott's questions were excellent. And one of the things that we do know is that if a screw is contaminated prior to going in, that contamination or that colonization is impossible to get out or impossible to kill by throwing in vancomycin on top. There's an innate amount of circulating bacteria that, of course, as Scott was alluding to, are then driven towards a new surface and form a biofilm. So the important part about clots is that the clots will continue to work after they've been implanted in the body and will continue to not allow bacterial colonization onto the pedicle screws. So what does that mean? That means that that 1 nanometer or 2 nanometers of biofilm will not be able to be formed. Will it actually treat an infection, like as if you had put it like gentamicin on a nail and then put it in? No, it's not an eluting technology. It's a non-eluting technology. So it simply keeps bacteria from attaching to a screw or a metal surface. And this, allowing this to happen, then can reduce biofilm, can then reduce micromotion, and can reduce possibly pseudoarthrosis.
Well, Celeste, that was an incredible discussion, and you're helping me work on my PhD with that kind of answer. Thank you so much. And David and Audrey, you know, I'm sorry to say I'm not a mechanical engineer. So this is not easy to understand stuff, but very worthwhile to understand. Mike Sherman, let's turn it over to you and your thoughts and questions.
Well, look, the problem of infected metallic hardware is massive, giant, and the economic impact is incredibly significant. And this question of biofilm is really at the heart of your ability to impact it. And I want to make sure I understand. Are you saying that your treatment can prevent the formation of biofilm?
Our treatment, I mean, our treatment is going to prevent the bacteria from entering the body on the implant, which is where I believe most of it comes from the studies that we've presented. Which I'll leave it to Celeste to talk about how that forms the biofilm.
Yeah, I'm not sure the bacteria form. I'm not. Well, I don't even know the answer to this. Maybe Celeste, you can tell me. Does biofilm form on all implants, whether they're infected or not, or does it take a microbe to initiate the formation of a biofilm?
Biofilms, in some form, are, I guess, form around any implant, any foreign body, right? And bacteria are attracted to that slime layer, as we all call it, but certain surfaces are more likely to get biofilms. We know hydrophobic surfaces like PEEK implants always have a very thick biofilm around them. You know, it was always one of the issues with pacemakers, things like that. Biofilm has its own issues, but then when bacteria colonize into a biofilm, they cause, as Scott was alluding to before, to get thicker and then become more robust with inflammatory factors, and therefore become more problematic for the implant.
Okay, so thank you, because so if the implant never so it's a race, right? It's a race in the formation of a biofilm layer, which doesn't exist when you take the part out of the package, and whether it gets finds an offending microbe. But your proposition is that there's no offending microbes anywhere near this implant.
Correct. That whole idea of race to the surface is, I did not coin that, but it's a very common discussion in biofilms, is that if the bacteria are already on the pedicle screw going in, nothing that you can do, whether you give oral antibiotics or put Vanco in your wound prior to closing, nothing's going to get them off the screw, and they're going to colonize and be there. If you've treated your screw prior to going in with a quat type technology, then the bacteria can never actually attach to the screw. And that's the important part, is they can't attach, and it's not a one-time only type thing where a lot of previous attempts have been if the bacteria came into contact with it, then you ruined that part of the coating, right? So it only worked once. This will continue to work. So it's not like it wears out or wears off. It's bound to the surface. It's not gonna come off in any way. It doesn't wear out in any way.
Okay, well, that's great. Thank you, Celeste. How hard, this is probably for David or maybe you, Celeste, how hard is it to treat? I mean, is it a batch process? Is it a vapor deposition process? Can it be done in the operating room? How does this work?
Yeah, we're setting up a production line for it. It's a chemical process. It's a 3-step chemical process, from adding the phosphonate layer to the polymerization step where we add the molecule that kills it. We're setting up a proprietary line in Eastern Pennsylvania. We're doing it in equipment. You're probably familiar with crest lines that are used to clean orthopedic implants that go through manufacturing. So we're setting it up in a line there. And so companies would send us this, much like you would send it to get an HA coating or a plasma coating, and then we will treat it, sterile package it, and then send it to the sterilizer to be sterilized and then returned to the manufacturer.
All right, well, look, congratulations. I've been aware of this technology for, as we talked about earlier, quite some time. So it's good to see it starting to approach the market.
Well, what an opportunity we have. Infection control is an area of such great financial cost and risk. In fact, you should know that about 1,400 patients die a day in the United States from sepsis. So infection control is a real problem. And just as a quick fact, does anyone know who the father of infection control is? Ignaz Semmelweis, a Hungarian-born scientist. I'll leave that name for you to research. And as you're researching that, let's consider and take the time now to ask you, our audience, what do you think? What do you think about this company? Is it a game changer? Do they have a game-changing technology? Can David make it happen? Will they challenge the standard of care? To answer these questions and address your thoughts, let's turn it over to Robin Young, publisher of Orthopedics This Week, who will present our poll. Robin, over to you.
Thank you, Tony. We polled our live audience, and here are the results. 9% of our audience think infection-proofing medical implants is a winner and game changer. 47% think OrthoBond's technology of making every implant bacteria-free could challenge the standard of care, which is currently prophylactically drenching the surgical wound with such antibacterial products as vancomycin. And finally, 44% think this is a promising technology but requires more work. Perhaps these audience members took note of OrthoBond's regulatory pathway. I hope you enjoyed this presentation, and if you want more information either about OrthoBond or how to be part of the live audience for the pitch, or even if you want to present a new technology or company, go to our website at Orthopedics This Week. And finally, if you like this view into the future of medicine, which is changing at an ever-accelerating rate, then let us know by clicking on the like button, of course. Thank you very much.